we are reducing oxygen. And so once you add those electrons, that oxygen becomes water. And then energy is going to be released through a tail-end process at the end of cellular respiration. Okay. Oh, and also I mentioned, so because you're removing these electrons are going from a high energy state in glucose. And so by breaking down glucose, you're lowering the delta G. So remember, anytime you lower the free energy, you're going to be releasing energy. And again, that's another reason why cellular respiration is going to be an exergonic process. And we mentioned that these oxidation reactions, these are going to be actually interactions between a fuel and oxygen. So anytime you have an oxidation, you're going to have oxygen present. And so here's our oxygen, and then glucose is serving as our fuel. And again, glucose serves as a fuel just like, octane would serve as a fuel. It's got lots of potential energy, and as it's broken down, it can be used to power chemical reactions. Okay? Okay, so remember I mentioned these little taxi cab drivers that are going to shuttle these electrons around? All right, now we're going to introduce those. So, whenever we take these electrons away from glucose, all right, and their destination is oxygen. So we have what are called electron carriers. And these electron carriers serve as these kind of little taxi cabs that shuttle these electrons from place to place. So we're taking these electrons away from glucose, and instead of giving them directly to oxygen, we're going to have these electron carriers that are going to help carry those electrons. So once the electrons are removed from glucose, they're going to jump in the cab, they're going to jump in the electron carrier, and it's that electron carrier that's going to take those electrons and then eventually give them to oxygen. And so there's different electron carriers, and over this chapter and the next chapter, we're going to run into 3 different types. The 2 that we're going to come in contact with today are going to be NAD positive and FAD. And so this NAD, this is nicotinamide adenine dinucleotide. I know, crazy name. But basically what this is going to act as is, again, this electron carrier. And so this is the oxidized form of this carrier. In other words, it's empty. It doesn't have any electrons in it. All right, so when these electron carriers are picking up these electrons, and again, these electrons are coming from glucose. So we're stripping electrons from glucose, we're loading them up in the electron carrier, and then that electron carrier is going to take it to oxygen. Well, when you add those electrons to these electron carriers, so if you're adding electrons, this is going to be a oxidation reduction. Reduction, right? Right, because you're adding electrons to something. So remember we said these electrons are going to come in the form of hydrogen atoms. And so once you add electrons to NAD positive, we say that it's going to be reduced to become NADH. And so you can tell if these electron carriers have electrons in them by whether or not there's an H present. And so those electrons are coming in the form of hydrogen because hydrogen has 1 electron, right? And it also has 1 proton. Same thing with this one. So this is another electron carrier, and it's going to serve the exact same purpose. It's a different molecule, but it's going to serve the exact same purpose of NAD positive. And so some of these other electrons that we're taking away from glucose, some of them will go, actually most of them will end up going to form this. And then some of them will also be added to this other electron carrier, FAD. And so when you add those electrons to FAD, again, it's going to be another reduction, and you're going to end up with FADH2. And so this NADH and this FADH2, these are going to help carry those electrons away from glucose and to oxygen. Now, you'll be hearing, instead of me saying NADH and FADH like 14 times, I'm going to call them NADs and FADs. Okay? Just in case you don't have one. So we got our NADs and we got our FADs. We've got the oxidized form that is empty. It's not carrying any electrons. In other words, the light on the cab is, bright. And versus, the reduced form, which is carrying the electrons. And again, you can tell that it's carrying the electrons because you've got some hydrogen atoms added to it. Does that kind of make sense? Okay. And so, and these NADs and these FADs, these represent, they have a lot of this stored energy. All right, and so it's these NADs and these FADs are going to eventually, a lot of the energy that gets transferred in cellular respiration is going to reside in these NADs and these FADs. So these NADs and these FADs, these electron carriers, have a whole bunch of energy stored in them. And so a lot of the energy that's going to be used in cellular respiration, or not used, but a lot of the energy that we're taking away from glucose are going to be carried in these electron carriers. And they're going to be carried in those electron carriers until they can turn around and give them to oxygen in order to form water. Okay, you're still okay? Okay. All right, so now we need to introduce the concept of the electron transport chain. And I think we've mentioned it before. But the electron transport chain, this is where these NADs and these FADs, these electron carriers that we've generated, this is where those NADs and those FADs, they're going to come into the inner mitochondrial membrane. So we've got our FADH2 and our NADH. And so here, remember the mitochondria, it's got those double phospholipid membranes, right? And so cellular respiration is mostly occurring on that inner mitochondrial membrane. So that's what this is right here, the inner, the most internal membrane of the mitochondria. And so within that membrane, you've got all these proteins. It's a series of proteins. And these proteins are going to make up the electron transport chain. And so what's going to happen, all these electrons that you gathered from glucose that are being carried by these electron carriers, all of these electrons are now going to be dropped off to the electron transport chain. So if we're removing electrons, we're donating those electrons, if we're letting the electrons get out of the cab, so to speak, all right, it's becoming oxidized. So once these electron carriers donate their electrons or give their electrons to the electron transport chain, these guys are going to go back to their oxidized form. So these will be converted back into FAD and NAD positive. Remember we said the oxidized form tells you that there's no more electrons there versus the reduced form. So you're taking these electrons away from glucose. All right, once you remove them from glucose, you give them to the electron carriers. And then those electron carriers go to the electron transport chain. And so once they reach the electron transport chain, the electrons have reached their stop, so to speak. And so those electrons will get out of the electron carriers, and they're going to go down this electron transport chain. And at the bottom of the electron transport chain, you're going to have what's called a final electron acceptor. And so in this case, in cellular respiration, in aerobic respiration with oxygen, this final electron acceptor is always going to be oxygen. So as all of us are sitting here loving life and loving cellular respiration and biology lecture, our cells are breaking down glucose and making energy. And so those electrons that are being added to oxygen are going to eventually reach oxygen, and when you add those electrons to oxygen, that's what's going to give you water. So the electron transport chain is where we get water as the product. And so this water forms as a result of those electrons that were taken away from glucose, and then we give them to oxygen by these electrons moving down the electron transport chain. And so we have these electron carriers. They've got their electrons. They're going to the electron transport chain. And so what's going to happen is these electrons are going to move from one protein to another until they reach the final electron acceptor, which in this case, in aerobic respiration, is always going to be oxygen. And so once those electrons meet up with oxygen, they're going to form water. And so this process is also going to be exergonic. Remember, what do we say about oxygen? Right? What's its main characteristic? It likes electrons, right? It's electronegative, right? So it hogs those electrons, it hogs that negative charge. So these proteins, and these proteins are actually arranged in a way which as you move down the proteins, you're actually decreasing the free energy. In other words, this first protein in the electron transport chain, it's going to have a higher delta G than the last one. So as these electrons are being passed from protein to protein, they're moving to a lower energy state. They're decreasing their delta G. Remember, anytime you decrease your delta G, you're going to have an exergonic reaction. And so another reason why cellular respiration is an exergonic process is because of this electronegative oxygen. Remember, oxygen is pulling electrons towards it. It's like a magnet for electrons. And so these electrons are getting happier, the closer they get to oxygen. And so a happy electron is one that has a low delta G, right? So these electrons are decreasing in free energy as they're moving down the electron transport chain. And then once they meet up with oxygen, you're going to get the production of water as a product. All right, and so we're going to go back. This was just kind of an introduction, but once we start getting into the details, we'll talk about this a little bit more. But I just want y'all to understand kind of the concept of the electron transport chain. So it's there for the electron carriers. It's basically like a pathway for the electrons to meet up with oxygen too. And so you've got these electrons, they're being carried by these electron carriers, and then once you've reached the electron transport chain, they'll jump out of the cab, they'll go down the proteins, decreasing in free energy, becoming more stable, and then meeting up with oxygen to form water. All right, so at this point, we've covered this part of the equation, or at least the introduction of this part of the equation, right? Taking electrons away from glucose and giving them to oxygen. So the flow of electrons, so to speak, is you're always going to start with these electrons at glucose. And then those electrons, they're being removed, and so they're then going to be carried by these electron carriers. And then from those electron carriers, they're going to be given to the electron transport chain. And then as they go through the electron transport chain, those electrons will finally end up with oxygen. So this is the flow of electrons. Flow of electrons in cellular respiration. All right, so are we okay with all of this? Questions, comments, concerns, thoughts? All right, so this is kind of what we were just talking about, this flow of electrons. And in general, these electrons are going to go to these electron carriers. But the reason why it has NADH right here is because this is going to be the predominant electron carrier that we're going to use. But we're also going to use our FADs too, our FADH too. But the one that's going to be used more often is going to be this NADH. And so again, we're taking these electrons away from glucose, we're giving them to these electron carriers. Then those electron carriers are going to drop those electrons off at the electron transport chain. And then they're going to meet up with oxygen. And then once they meet up with oxygen, that's what's going to give you water. And so again, this is what's going to give us water in the product of the equation. And again, because you're breaking down glucose, because these electrons are going from a high energy state to a lower energy state, this entire process is going to be exergonic and is going to be releasing energy. Okay, so some of y'all might be wondering, maybe not, but some of y'all might be wondering, well, why do you have to go through all of this complicated process just to give some electrons to oxygen? All right? Well, the reason why is because if you just took those electrons away from glucose and immediately gave them to oxygen, you would release so much energy. There would be like an explosion of energy. And so much so that it's hard for the cell to adequately collect and utilize all of that energy. It's just so overwhelming for the cell, it's just not going to know what to do. It's like, oh my God, I have all this energy. What's happening? All right? And it's not going to be able to efficiently collect and utilize that energy. So instead of just having a wow, bam, thank you, ma'am reaction where you get it all at once, this is why we use the electron transport chain. And so we create this like stepwise fashion of slowly introducing these electrons to oxygen. And so the electron transport chain, because these electrons are gradually decreasing in free energy, becoming more stable, you get this release, this manageable release of energy. And so by making a little bit of energy at a time, your cell can adequately, collect it and manage it and utilize it. Versus if you just have this explosion of energy, a lot of that's going to be lost and you're not going to be able to utilize it. So we evolved the electron transport chain to help in this process, to slow down this process so that you can collect energy in a manageable fashion. And so again, since we're taking these electrons, they're going down the electron transport chain, and then those electrons are going to meet up with oxygen in order to form water. Okay? Okay, so now it's time to get into cellular respiration. All right, so cellular respiration is made up of three primary processes. You've got glycolysis, you've got the Krebs cycle, which is the same thing as the citric acid cycle, and then you've got this oxidative phosphorylation. All right. So, the purpose of glycolysis and the Krebs cycle, in general terms, in big picture ideas. All right, the purpose of glycolysis and the Krebs cycle, this is where you're oxidizing your glucose. So both of these processes are used to strip those electrons away from glucose. And so, like we said, their primary function is to oxidize glucose. So in glycolysis and Krebs, all we're doing, we're stripping those electrons away from glucose. All right, so that's the purpose of both of these. And then the third step, this oxidative phosphorylation, is actually going to have two processes that work together within it. The first one is going to be this electron transport chain, or the ETC that we just talked about. The second step is going to be a process called chemiosmosis. And so what chemiosmosis is the definition in general terms is using some sort of gradient. Remember when we talked about membranes, we had active and passive transport, we had with the gradient, we had against the gradient. So same sort of thing. So chemiosmosis is using a gradient of something in order to power something. All right? In this case, in chemiosmosis, we're going to use a gradient of hydrogen ions, or protons. Those protons are going to form a gradient across the inner mitochondrial membrane. And so that gradient is then going to be used to make a whole bunch of ATP. So we talked about kind of where all the products of cellular respiration come from, except for ATP. Well, chemiosmosis, its main goal, this is where you're going to get most of your ATP. And now, the electron transport chain, this is going to be where you are giving those electrons to oxygen in order to make water. Alright, so in general terms, glycolysis in the Krebs cycle are breaking down glucose. They're oxidizing glucose. Whereas in oxidative phosphorylation, in the electron transport chain, this is where those electrons are meeting up with oxygen to make water. And in chemiosmosis, this is where you're going to generate all of your energy. Alright, am I going too slow or too fast? I was purposely trying to go a little slow because my boss was here.
No, I'm getting it.
Are you good? Okay. Are you falling asleep because you're getting it too low?
No.
All right. Okay. All right. So, again, big picture ideas, right? So, we're going to start with glycolysis. And glycolysis is going to start this oxidation of glucose. Now, glycolysis, by definition, glyco means sugar, and lysis means to break. So we're literally breaking a molecule of sugar in half, in this case, glucose. And so we're